So, welcome all. Today we are going to study about the plant tissue system. And as the name suggests, we have to study how the building blocks of the plants in our portion can be built by these tissue system. So, first of all, now we have some aims and objectives to fulfill the study of plant tissue system. Like we have to study what are the types of plant tissue system, understanding of temporary and permanent tissue. These are the two broad categories that come under the plant tissue system. Temporary plant tissue system and permanent. As the name suggests, the temporary one. Basically, the meristematic tissue which present in a very first stage of the plant development come under the temporary tissue system. Secondly, the permanent tissue. As their name suggests, they are the permanent tissue throughout the life of the plant. Now, we also have to see what kind of cells are present inside these particular kind of tissues. So, let's start with the introduction part. So, firstly, we are going to see here that what are the tissues. So, as before going to the tissue part, we have to know that the tissue are basically the specialized aggregation of the cells which perform a special function together. So the collaboration of all the cells in a one particular platform and functioning out something that is called the tissue. Now you can also see here there is a diagram of one cell and another diagram of the tissue. So if you wanted to know more about the tissue, you also have to be very particular about what kind of the cell is forming that tissue. Because every tissue which present inside the body, they have a special role to play. And performing their special role, they need some special kind of cells inside them. So here you can see the fundamental, the cells, which are the fundamental and basic functional unit of any organism. And these cells are together will perform the function for the tissue. So if we see what kind of the tissue is present inside the plant body, now we have to go through this classification pattern of the plant tissue. You can see here, the very first plant tissue system is divided into the two broad categories, meristematic tissue system and permanent tissue system. As the name suggests, the meristematic tissue system, they are actually the kind of the temporary tissues. Okay, these are the type of temporary tissue which do not present inside the plant body for their total lifespan. They just have to be present in the plant body for very initiative and first step towards the plant growth and development. Hence, these meristematic tissues are only we can call them that they are the temporary tissue which present inside the plant for a temporary reason. Second, on another hand, these are permanent tissue. They are the mature cells and capable of division. They are not just present for the very short time period. Basically, in opposite, they are the cell which have a long life inside the plant body, and they are the also the mature cell. They they have capacity to attain maturity inside them, and also they cannot incapable, or you can say that they are incapable of the cell division. So basically, in this kind of tissue, the permanent one, cell only divide at their younger stages. They cannot divide up to when they get mature. So if we see the further fragmentation of the permanent tissue, here meristematic tissue majorly do not have any subcategories in them because these tissue do not have that much life to basically divide into the further integration. But as a permanent tissue, which have to present throughout the life of the plant, so that's why we divide them into the two broad categories. First category here is a simple, second one is a complex. So as the name suggesting them, they're the simple tissue. They are kind of very made up of very simple type of the cell. They do not have very special type of cell formation, and also They do not have any special kind of function to perform inside the plant body, but they are the building blocks of the cortex region and the very outer protective zone epidermis. So you cannot say that epidermal tissue or your simple tissue, they have a less function to perform. They are important to the cell, and but their function is little similar to each one of them, like parenchyma, collenchyma, sclerenchyma. They all have the almost similar kind of function to perform in the plant body. Their shape, their size, they do not vary that much as we will see in the complex tissue. So basically, when we are talking about the complex tissue, their name is the complex one. So that's why the function which has been dealt or can be done by the complex tissue, that is a little complex as comparison to the simple tissue. How? Like under these tissue, or mostly these tissue are made up of more than one type of the cell. So here, the in simple tissue, all the cell, the parenchyma, collenchyma, sclerenchyma. If this is the parenchyma tissue, the all the cell which made the parenchyma tissue is only parenchyma cell. Same in collenchyma. Whatever the collenchyma tissue or present anywhere in the cell, they all made up by the only collenchyma. Same to the sclerenchyma, they are also having or made up of the same kind of cells. But in the complex tissue here, xylem and phloem, these are also two different tissues, but they are made up of different kind of cells. You can see xylem have the vessels, xylem have tracheids, parenchyma, fiber. On other hand, phloem also have these kind of differentiation into their cell pattern. They have the sieve tubes, companion cells, and phloem parenchyma and the phloem fibers. So these tissues have a major broad category of the differentiation. But on the other another hand, the simple tissue do not perform any kind of variant or much diversity as you can see inside the complex tissue. So here you can see a diagram of the meristematic tissue. This is a very simple kind of the cell arrangement. They are just have all the cells are identical. Same kind of cells are present here. Only they are having one cell wall, cell membrane, centrally placed nucleus, and the cytoplasm. So these three things are the basic fundamental units which present in the meristematic tissue cells because they do not need any kind of very complex organelle inside them because all they need to do a single work of the cell division. They do not have to play a very major role inside the meristematic tissue group. That's why these cells are simple and perform just one function, and that is cell division. We can also see some very basic features which come under the meristematic tissue, like they are composed of cells that divide continuously. Okay, so this is a major effect you can see under the meristematic tissue. That the major work they perform there is just for the dividation. No any other can be done by the meristematic tissue. They only work for the dividation of cell or cell division. Second, if we talk about their structure, so they can be spherical, they can be oval, polygonal, or rectangular in shape, and their cell wall. The cell wall is very thin and made up of the cellulose. So this is a region here, like why the cell is thin and made up of cellulose. So basically, if they have to do cell division process, so for any cell, if they wanted to do the cell division process, they need to go through either two diversion, or you can say there are two processes which a cell have to follow for completion the cell division process. Like process 1, that is the karyokinesis process. Process 2, that is the cytokinesis process. So in the process 1, that is the karyokinesis process, plant cell have to go through to the division of the nucleus. second process, that is the cytokinesis, cell have to go through to the cell wall, or you can say the cytoplasm division. If the cell have a thick cell wall inside them, so there is a complexity will be increases for the cytokinesis process. So that's why these meristematic tissue or their main core function is just to perform the cell division. So that's why, hence, they need a cell wall which is thin and easily breakable into the two components. So that's why these meristematic tissue have a very thin cell wall. These cells are closely arranged without intracellular space, so actually they divide and place just right after to each other. There is no intercellular space present. So I think you all know what is intercellular space. Intercellular space is a gap between two cells. It is not present inside the meristematic tissue. Cytoplasm, if we talk about the cytoplasm of meristematic tissue, this is abundant and nuclei is large. So basically, a large nuclei help to easy the karyokinesis process. Means karyokinesis means division of nucleus. So, if the nucleus is large, so they can easily divide and form the two cells. Vacuoles, on the other hand, they are absent, and if they are present, they are very few. So, why the vacuole are absent in the meristematic tissue? Actually, what is the work of vacuole to do inside a cell? The only work which vacuole perform inside the cell is it for only the storage purpose. But we know the nature of meristematic tissue. They do not need any kind of storage. All they just wanted to do is division. So why they need to storage something when they do not have to live long? So that's why they do not have the vacuole inside them, or if they are having a vacuole, the numbers and the size of vacuole is very few or very small. So these are the basic characteristic feature which can be seen inside the meristematic tissue. Now there are some positions in the plant at which place you can see the meristematic tissue present. Like in the very first here, you can see the apical meristem. Okay, this is a top version, and they are having the meristematic tissue for the length purpose to increase the length of the plant. Second one you can see here, this is the intercalary meristem. Intercalary meristem basically present for the internodal growth. Okay. This is the internodal portion. Two nodes are present here, and the difference between, or you can say, the distance between two nodal portion is called as the intercalary meristem. At this position, a number of meristematic tissue present, and last but not least, the lateral meristem. This lateral meristem actually have a great tendency to present inside the dicot plants, because the dicot plant have shown the secondary growth which basically help to increase the girth of the plant, the thickness of the plant. So this thickness can be help to grow the plant laterally, and this lateral meristem basically help to grow the girth of the plant by presence of the meristematic tissue. So these all the tissue plays very important role in each and every kind of the development inside the plant body. If we talk about the meristem classification, then the apical meristem, as you seen in the diagram, they are having the position at the top of the root tip and shoot. So it can be present any double portion, it can be root and it can be shoot. And their function is increase in length or the axis. Second is the lateral meristem. So basically, on the lateral sides of the root stem, they are present, and their function is to increase in diameter of the plant. As I already told you in diagram, all the diameter portion will be, you can say, diameter portion has been increased by the lateral meristem. Now, third is intercalary meristem. So this present in between the bases of the leaf and also the internodal portion. So, their major function of any meristematic tissue is basically increase the number of the cell, or it can result into the length of the plant and the girth of the plant. So, same here, function is to increase the length of the plant of its body. So these all meristem has a main function to perform just to increase the length and the girth of the plant. You can also see a section. Okay, this is a LS, the stem apex and the root tip showing meristematic tissue. You can see there are several simple cells are present here. They are not the complex cells, they are very simple kind of cells, all almost identical. And you can also see there is no intercellular space present in any of the cells. So hence proven that they are the meristematic type of cells. Now the function we already see, the meristematic tissue continuously divide and help to form the new cells, and also they will help to form the permanent tissue. So when a optimum length has been occurred by a plant and they do not need to divide furthermore, at that time this meristematic tissue basically converted into the permanent tissue, or they can you can say that these are the very first tissue from the permanent tissue. Next, you can see here. Now, as these meristematic tissues started the growth of the permanent tissue, so when the cell formed by the meristematic tissue takes up a specific role and lose the ability to divide. So, initiation of the permanent tissue, basically what happened here, when a meristematic tissue convert into the permanent tissue, the very first thing they have to lose their specific role, that is ability to divide. Now, at this stage, plant cannot divide, they cannot make new cells. So as a result, they form the permanent tissue because they cannot divide now. So as they are permanent tissues, permanent cells, hence we can call them the permanent tissue. This process of taking up a permanent shape, size, and function is called as the differentiation. Now, at this position, plant have to be perform a very specific role now. So they need to stabilize their cell. They need to stabilize their shape, their size, their function. And on the basis of their division, on the basis of their shape, their size and their function, this whole process is basically called the differentiation process. So cells of meristematic tissue differentiate to form different type of permanent tissue. So basically, as if we are talking about a meristematic tissue, they are all the same kind of tissue. But when they led by the not the process of division, but when they led by the process of differentiation, they can be chosen to have a different shape, different size, and different function. Now, if we see the classification of the permanent tissue, you can see there are the two broad categories here. In the case of permanent tissue, the simple permanent tissue and the complex permanent tissue. In simple permanent tissue, we are having here parenchyma, collenchyma, and sclerenchyma. On the other hand, in the complex permanent tissue, we are having the two cells, xylem and phloem. Now we will see how these cells specifically work. So initially, if we talk about the simple permanent tissue, what happened here? They are basically made up of one type of the cell, and that form the homogeneous or uniform masses. So basically, all the cell which is present inside the simple permanent tissue, they are having the same kind of structure and function. So basically, when we are calling them the homogeneous, so on the basis of that, similar structure not, we can divide the plant into the two broad categories. The plant cells can be the homogeneous cells, and plant cell can be the heterogeneous cell. So In the case of homogeneous cell, all the cells are alike. Okay, means all the cell have a same structure, same color, and same function. If we are talking about the any cell which is heterogeneous in nature, so at that time cell have a different kind of structure, cell have a different type of function, and cell may have different type of shape. So all these things, if they are different, we can consider as a heterogeneous tissue. But if they all are the similar one, we can call them a homogeneous tissue. So our permanent tissue, our simple permanent tissue, basically include these three tissue system: parenchyma tissue system, collenchyma tissue system, and sclerenchyma tissue system. Now, the very first cell of the simple tissue system, we will take it as a parenchyma. So these cells, if we watch their characteristic feature, if we see their characteristic feature in the parenchyma tissue, you can see that the cells are isodiametric, means they are equally expand on all sides. Okay, meaning of isodiametric, that if a cell can be expand similarly in all sides, we can call them the isodiametric cells. They may be oval, round, polygonal, or elongated. So there is not a definite kind of shape you can tell about the parenchyma. So they especially vary according to species, according to their function, and according to their part in which they are present. So they can be oval, they can be round, they can be polygonal, and they can be elongated, a little long. Nucleus is present and hence living. These are the, you can say, these are the only cell in the simple parenchyma, simple tissue, which are living cells. Okay, no other cell like the, we will study the collenchyma and sclerenchyma after this parenchyma tissue, and you will see there that there is no cell living. Still, only just this parenchyma, the living one. So the cells wall are very thin and made up of cellulose. So they having a very character of like the meristematic tissue also. They're having the simple thin cells and they are the living cells, and also their cell wall is made up of the cellulose. Cytoplasm is dense with a single large vacuole, but this character is different from the meristematic tissue, that their cytoplasm is dense and they are having a large vacuole. If you see the function wise, parenchyma have to perform the function of storage at very vast level inside the plant. So majorly when we are talking about any storage material inside the plant, the first name that come under the mind is the parenchyma tissue. So these parenchyma tissue have major role to play for the storage purpose. So here, that's why we are having a large vacuole system inside them, because what the function of vacuole do inside a cell, that is a storage function. So that's why they are having a large vacuole. Intercellulose waste may be present. So this is also certain that maybe they are having intercellulose waste. So it is a maybe situation. May be present or maybe not. That depends on the species. May contain chlorophyll. Parenchyma which contain chlorophyll are called chlorenchyma. This is also a special feature of the parenchyma. Like some of the cell of parenchyma, they have present chloroplast inside them. If the chloroplast is present inside the parenchyma cell, so what they have to do? They need to do the photosynthesis. So at that time, we called these parenchyma as a chlorenchyma because the chlorophyll is present. So that's why we named them the chlorenchyma cells. There are several other type of the parenchyma also present in several group of the plants, like these erenchyma. So basically, if we are talking about the parenchyma, means there are the parenchyma which have a very large air chambers. Okay, if a cell having a large air chambers, we can call them the parenchyma. Basically, this parenchyma thing is a very important part of the hydrophytes plant. I think you know the hydrophytes plant. These plant which present inside the water. If any plant which is present inside the water, they need to, the very special need of their plant is to get the air. As most as the air they can store, this will be beneficial for them. That's why they convert their parenchyma tissue into the parenchyma. So basically, parenchyma store a large amount of air for them to their survival. Next is here you can see the chlorenthyma, all the green cell you can see here. Okay, so basically these green cells, they are the parenchyma initially, but after having this green chlorophyll inside them, they will be known as the chlorenthyma cell. But initially they are the parenchyma. Occurrence means where the parenchyma tissue is present. So basically they present in the cortex of root, ground tissue in stem, and mesophyll of the leaves. So these are the very, you can say these are the very spaces where you can find the parenchyma inside a plant body. Function, if we talk about the function of parenchyma, they store and assimilate food, giving mechanical strength by maintaining turgidity. So this giving mechanical strength. Basically, this is a very important function that is done by the parenchyma because all the epidermal tissue system, like presence of epidermis, that is the outermost layer, having followed the hypodermis, these all things, what they made up of? They actually made up of the parenchyma cells. So this parenchyma basically have to perform a function to give the mechanical support for the growth of the plant. Prepare food if chlorophyll is present. So as we just see that the some parenchyma have the chlorophyll inside them, and if they are having the chlorophyll inside them, so their major function is to perform the photosynthesis. So if they are doing the photosynthesis, hence they can do the food preparation work for the plant. Store waste products like tannin, gum, crystal, resin. So there are many excretory products which has been secreted by the plant body. So these all secretory things can be stored by the parenchyma in the form of tannin, gum, crystal, and resin. So these are the all four very important functions in the plant body that is performed by the parenchyma cells. Next is our cell is a collenchyma. So these cells are elongated and are circular, oval, polygonal in cross section. So they are also like parenchyma. They can be elongated, circular, oval, and polygonal, depend on the which area they are present. Cell wall is unevenly thickened with cellulose at the corner against centrocellular space. This is a new feature, or you can say this is a little modification of parenchyma, that is a collenchyma. Means you can see here, if this kind of thickening, okay, this is a thickening, you can see here a little thickening on the cells here. So these thickening basically a characteristic feature of the collenchyma tissue, but they do not have an even thickening. Means the whole and total a cell wall do not have even thickening. Means same amount of thickening is not present on the entire wall. Only at some point there is a thickening present, and some another point there is not exact same thickening has to be present. That's why we call them, they have the uneven thickening. Nucleus is present, hence this tissue is living. Tissue is living only at the initial stages when the thickening do not increase. Up to the level of thickening increment, if the thickening does not cover the entire cell wall, that time till the collenchyma tissue is living. But when the thickening covers almost all the spaces, most of the spaces, then at that time cell becomes start becoming dead tissue. Nucleus is present, vacuoles are small. That's why the vacuoles are the small in collenchyma, because their major function is not for the storage. Their major function is to give the mechanical support to the plant. Because of having this deposition on their cell wall, they actually strengthenize the wall of the plant and wall of the cell also. These strengthened cell wall make them a protective zone for the plant. Intercellular spaces are generally absent, so if we compare them to the parenchyma, the most of the intercellular space in the between the cell they are absent, or sometime a very few intercellular space present, but all the time mostly they do not have one. If they contain chlorophyll, they are also known as chlorinchyma. This is also a very rare situation. Not every time the collenchyma have the chlorophyll in them. Very specific plant have this character that they are having this collenchyma having chloroplast inside them. But this is very common for the chlorophyll having in parenchyma. Parenchyma having chlorophyll and called chlorenchyma, this is normal. But collenchyma having chlorophyll and called chlorenchyma, this is a rare character. Occurrence, if we talk about the occurrence, so mostly found under the skin. So that's why they can protect us. So that's why they just below present below the epidermis. Okay, epidermis is the outermost layer. You can see the diagram here. This angular thickening. Okay, not whole cell have been having thickening, but several spaces they have the thickening at some places only. Also here you can see after the epidermis and before the cortical regions there are a zone, a little darker zone. You can say the hypodermis. This hypodermis basically made up of the parenchyma here. So that's why chlorinchyma, because of this thickening pattern, they can give the mechanical support to the plant. Function, provide mechanical support to the stem. Being extensible, these cells readily adapt themselves to the rapid elongation of the stem. So actually these are very extensible cells. They have this extension process and these cells readily adapt themselves to rapid elongation. So if you wanted to do any elongation process in just below the epidermal portion, they are the cells you can work with. Last cell of the simple tissue system is the sclerenchyma cells. So these cells, basically, if we talk about their nature, they are long cells as comparison to the parenchyma and collenchyma. They are not very oval in shape. They are the long, narrow, thick, lignified, and usually pointed at both ends. Okay, these are the basic characters. They are long, narrow. Narrow means they lumen. This is called lumen. This is a narrower in the case of the sclerenchyma and usually pointed at the ends. Means when the cell have getting an end, they are mostly pointed. Their cell wall. This is the cell wall like they all are lignified. Chlorinchyma, they are having lignification at only just several points. Okay, they do not have the entire wall deposition. But in the case of sclerenchyma, this is what you can say modification of chlorinchyma. They have the thickening all around the cell. The cell wall is evenly thickened, okay, thickened from everywhere with lignin, and sometimes it is so thick that the cell cavity or lumen is absent. And sometimes this deposition have a very wide range, even this much range that they properly absent this lumen. Okay, the thickening take over the lumen spaces. So at that time, lumen got absent. You can see here. Okay, in this cell, there is only left is a thickening. There is just a very tiny lumen have here. So that's why their nucleus is absent. Hence, the tissue is made up of dead cells. So that's why they are having so much thickening on their cell lignification. So that's why their nucleus is not going to work and they're absent. So that's why hence the cells are dead cells. They have simple often oblique pit in the walls. Sometimes they have the pits for the transportation. Okay, but this is rare, not every time they have pit. The middle lamella, the wall between adjacent cell is conspicuous. So this middle lamella, okay, this is a middle cell between two. adjacent cell is conspicuous, not very clear to be identified, and they do not have any intercellular space present in them. Okay, there is no intercellular space which can you find inside the sclerenchyma. Now, if we talk about their occurrence, how or at what place they are present. So basically, they found abundantly in stem of the plant like hemp, jute, coconut. So mostly that any part which is very hard, okay, for give the major mechanical support to the plant. At that places, the sclerenchyma tissues are present abundantly. Like it is present in stem around vascular plant and the leaves, hard covering of seed and nut. So these all things which you are seeing here, these all places are basically at that place where plant needed to be very strong. So monocot stem, you can also see they are having in monocot stem after the hypodermal region they are having a very dark thickening of the cell. So, due to their evenly deposition of the cell wall, you can easily identify them from any other tissue. Function. So, there is two function basically which performed by the sclerenchyma cell. Very first, they give mechanical support. This is a major function that has been done by the sclerenchyma cell. They give the rigidity, flexibility, and elasticity to the plant body. So basically, they protect the plant. They protect any kind of harm from the outside to the inner side. So this is a very important role that these tissue present inside the plant body. You can see here they are having this thickened wall with a very reduced lumen inside them. So these are some fiber and sclerates, which also the part, or you can say these are also the type of the sclerenchyma. They have the same kind of properties which are present inside the sclerenchyma tissue. They are also having a very short lumen. They are the elongated cells and highly thickened, and they also perform the function for the mechanical support and elasticity of the body. So you can see the sclerenchyma cell are usually elongated with tapering ends. Such cell are commonly called as a fibers. So if the sclerenchyma cell which is elongated long with tapering ends. So if any kind this kind of combination you found at anywhere, you can simply call it the fibers. On the other hand, the cells which are little shorter and irregular in the shape, such cells are commonly called as the sclerates or stone cell. For having a stone cell or sclerates, cell need to be shorter and irregular in shape. But in the case of fiber, cell needed to be elongated, but they are also having the tapering ends. So these two are the type of the sclerenchyma. There are some detailed structure of the sclerates has been given here, like most common in the fruit and seed, because they are very tough. So that's why we can call them the stone cell. They are very short, isodiametric in shape, spherical, oval, and also they have the same lignification process as happened into the sclerenchyma, and majorly their features and their function are way more similar. Like their function is also the protect the plant from the stress, strain, give mechanical support, writing of the pulp. So these are the things which have performed by the sclerates in a particular organs. So now this is the combined, you can say, a chartable account for the how the parenchyma, collenchyma, sclerenchyma work. Like you can easily difference out between them. You can also see the what are the common characters, like all the major events and all the major characteristics have been compared in this table, like cell structure, thin wall, thick oleate corners, sclerenchyma, they are thick walled. In the case of parenchyma, cell loosely arranged, cell compactly arranged, cell compactly arranged. So you can easily clarify that what are the basic differences between them or what are the basic thing. So this is a summarized version. You can see the parenchyma, collenchyma, and sclerenchyma has been presented in this. This is the diagrammatic representation of all the three tissues. You can be very clear. Uh, Although we already studied this individually, now you can see them and do the comparison between each other. These are also some, you can say, also modified version and diagrams. So now there are some questions you need to go through as you already studied about what kind of tissue system has been present inside the plant body. So I think you might be able to answer all these questions. I will upload these questions to the your elements and just go through these. And if still you are going to face any problem, you can just ask me. These are some references for the books and chapters that you can go through, and these are very reasonable explanation of all the type of the tissue system which is present inside the plant body. Okay, still if you have any confusion, you can come to me and study these paper and the book. Also, they are giving you the appropriate level of knowledge about the plant tissue system. Okay, so I think this is all for today.